The digestive system, Part 2. Different types of associated glands are salivary glands, gastric glands, liver, pancreas, and intestinal glands. These glands secrete digestive juices during the process of digestion. There are three pairs of salivary glands: parotid, submaxillary or submandibular, and sublingual glands. The parotid glands are located one on either side in the groove between the ramus of mandible and mastoid process below the ear. The duct of parotid, that is, the duct of Stenson, opens into the vestibule of the mouth cavity near the crown of the second molar tooth. The submaxillary glands are present medial to the mandible in the submaxillary triangle. The duct of the gland, known as the duct of Wharton, runs anteriorly and medially to open in the floor of the mouth. The sublingual glands are situated beneath the tongue. Each sublingual gland has about 10 ducts called the ducts of Rivenus, which open into the floor of the mouth. Histologically, the salivary glands are racemose glands and they look like clusters of grapes. Each gland consists of large numbers of acini, and each acinus is lined by pyramidal epithelial cells standing on a basement membrane. The function of salivary glands is to secrete salivary juice. The juice is conveyed by fine ductules, which unite with similar ductules from other acini to form bigger ductules. The latter unite to form the main duct of the gland. The salivary gland secretion is very rich in salivary amylase, a starch-splitting enzyme. The secretions of sublingual and submaxillary glands are, however, rich in mucin. About 99.5% of saliva is water. It contains two important enzymes, amylase and lysozyme. Amylase is starch-digesting enzyme which breaks starch into maltose and triose. Lysozyme causes lysis of several common bacteria that may be present in the mouth. Mucin in saliva helps to lubricate the food for swallowing. The saliva helps to wash out bacteria of the mouth, aids in speech, and also acts as a solvent of many substances. Such substances, when dissolved in the mouth and come in contact with taste buds, produce the sensation of taste. The glands that are present in the stomach are known as gastric glands. They are of three types: fundic glands, pyloric glands, and cardiac glands. Fundic glands, also known as oxyntic glands, occur in the body and the fundus of the stomach. These glands secrete hydrochloric acid, pepsinogen, and soluble mucin. The secretions are strongly acidic. Pyloric glands occur in the pyloric area of the stomach. The secretion is rich in mucin, but it does not contain hydrochloric acid and is slightly alkaline in reaction. These glands also secrete a hormone called gastrin. cardiac glands occur in the cardiac area of the stomach and secrete mucin and a little amount of pepsinogen. Gastric juices such as hydrochloric acid, pepsin and mucus secreted by gastric glands perform several functions. Hydrochloric acid is necessary for the conversion of pepsinogen into pepsin. It kills bacteria that are present in the food. The presence of hydrochloric acid makes the gastric chyme acidic in reaction. When the acidic gastric chyme enters the small intestine, it stimulates the liberation of some gastrointestinal hormones such as secretin and cholecystokinin or pancreazymin, etc. Pepsin breaks down protein molecules of the food peptones, proteoses and polypeptides. These partially digested proteins, when enter the small intestine, provoke the secretions of further gastrointestinal hormones. Mucus protects gastric mucosa against the probable injury by acid-pepsin mixture and other injurious agents. It also lubricates mucosa, which in turn assists the movement of food in stomach. It retains water within it so that the gastric epithelium keeps a watery environment. The liver is the largest gland of the body, weighing about 1.5 kilogram. It is located in the right upper part of the abdomen, just below the diaphragm. On the lower surface of the right liver lobe, a thin-walled pear-shaped sac is present called the gallbladder. The basic structural and functional unit of the liver is the liver lobule. It is a somewhat cylindrical structure, several millimeters in length and 0.8 to 2 millimeters in diameter. The human liver contains 50,000 to 1 lakh individual lobules. The liver lobule is constructed around the central vein and is composed of many hepatic plates that radiate centrifugally from the central vein as spokes in a wheel. A hepatic plate is 1 to 2 cells thick. Small bile canaliculi lie between the adjacent cells. The liver cells synthesize bile, which is first conveyed by canaliculi. The canaliculi join and ultimately form 2 hepatic ducts which meet to form a common hepatic duct. Bile is formed and delivered continuously via hepatic ducts. From the common hepatic duct, the bile flows into the gallbladder via cystic duct. Bile is stored in the gallbladder and delivered via bile duct. Near the duodenum, bile and pancreatic ducts join to form hepatopancreatic duct, which opens into the duodenum. The opening is guarded by a sphincter known as sphincter of Oddi. Liver acts as an exocrine gland for the secretion of bile and a storehouse for various substances like glucose as glycogen, lipids, vitamins and iron. Liver plays a prominent role in the metabolism of carbohydrates, proteins and fats, and regulation of blood glucose and lipids. Liver plays a protective role by detoxifying substances including drugs and alcohol. It helps in the removal of unwanted particulate matter from the blood through the mediation of macrophages. Bile salts are necessary for the digestion and absorption of fats. They activate pancreatic lipase and help in emulsification of food fats and their absorption in the intestine. They keep cholesterol in solution in the bile and have a mild purgative action. The pancreas is a gland partly exocrine and partly endocrine, but the main bulk is constituted by the exocrine part. The gland consists of a head, neck, body and tail. The head of pancreas fits in the C-shaped curve of the duodenum. The body of pancreas extends to the left and its tail touches the spleen. Its exocrine part consists of numerous round acini. The acini drain into small ductules. These ductules unite to form larger ducts and ultimately the main duct, which is known as pancreatic duct. The pancreatic duct unites with a common bile duct at its terminal portion, and the combined duct opens into the second part of the duodenum. The exocrine pancreas secretes enzymes that play a very important role in the digestion of carbohydrates, proteins and fats. The endocrine part of the pancreas is in the form of numerous rounded collections of cells that are embedded within the exocrine part. These collections of cells are called islets of Langerhans. Each islet is separated from the surrounding alveoli by a thin layer of reticular tissue. The islets are richly supplied with blood through a dense capillary plexus. There are three main types of cells in an islet: the alpha cells, the beta cells and the delta cells. The alpha cells form about 20% of the islet cells and secretes the hormone glucagon, which initiates the conversion of glycogen to blood glucose. About 80% of the cells of the islet are the beta cells, which secretes the hormone insulin. Insulin has an opposite action to that of glucagon. It helps in lowering down the blood glucose level by converting glucose into glycogen. The delta cells produce the gastrin and somatostatin. Their number is very small. In the islets of the human pancreas, the alpha cells are arranged towards the periphery or cortex of the islets. The beta cells near the center or medulla, and delta cells are also placed peripherally. Intestinal glands are present in the mucosa of the small intestine and are of two types: crypts of Lipocin and Brunner's glands. Crypts of Lipocin are simple tubular glands opening into the lumen of the small intestine. They are lined by columnar epithelial cells and contain some mucus-secreting goblet cells. The juice produced by these glands is known as succus entericus. It is a thin watery juice containing various enzymes like enterokinase, etc. Brunner's glands are compound tubular glands which are found only in the submucosa of the duodenum and open in the spaces in between the villi. These glands secrete alkaline mucus-rich juice. In mammals, there is much variation in the number of teeth in different species. but it is constant and characteristic for a species. The number and kinds of teeth in a mammal species can be represented by a sort of equation called dental formula. Since the two halves of each jaw are identical, only the teeth of one half are recorded. Thus, the formula showing the number and arrangement of teeth in one half of each jaw is called dental formula. The dental formulae of the upper and lower jaws are separated by a horizontal line. The numbers in the formula represent incisors, canines, premolars and molars in this order, starting from the middle to the backwards. The number of teeth shown in the formulae multiplied by 2 gives the total number of teeth in a species. The number of teeth according to the dental formula for a dog are 21. Thus, the total number of teeth in a dog are 21 multiplied by 2, that is 42. Let's take another example of the dental formula for a cat. Here, the total number of teeth in a cat are 30. The ruminant mammals such as cattle, sheep, goat, deer, camel, cow that chew cud have a compound stomach. A compound stomach is formed of 4 chambers: rumen, reticulum, omasum and abomasum. However, in some ruminants like camel and deer, omasum is absent. In fact, the first 3 stomach chambers are derived from the esophagus, and the abomasum is a true stomach. Rumen is the first and the largest of 4 chambers. In camels, both rumen and reticulum have pouches known as water cells which store metabolic water. This usual arrangement of the digestive tract in ruminants is an adaptation to digest the cellulose of the plant cells. None of the mammals are able to synthesize the enzyme that digests cellulose except ruminants which digest it by harboring cellulose-digesting bacteria and protozoa within their stomach. The cellulose-digesting microorganisms live in the rumen and the reticulum of the ruminant. The food swallowed first enters the rumen and then the reticulum, where microorganisms digest and ferment the liberated glucose, since conditions inside the stomachs are anaerobic. From the reticulum, the coarser plant materials are regurgitated for re-chewing and then swallowed again for further enzymatic action. The microorganisms use the products of their digestion and release fatty acids. The fatty acids move to the omasum along with some of the bacteria and protozoa. In omasum, the material is concentrated by the reabsorption of water through the omasum walls. Then it moves to the abomasum, where acids kill the microorganisms. In the small intestine, the amino acids, glucose, fatty acids and other monomers are absorbed into the bloodstream. Due to the presence of 4 stomachs, the digestive tract of a ruminant is more specialized than that of the other herbivores in case of 1. The digestion of cellulose, 2. Absorption and recycling of water from the enormous quantity of saliva used in the digestion by the microorganisms, and 3. in killing of microorganisms by acids.